So I'd like to go over this new paper from David R. Liu Lab, and it's in cahoots with the Mo Khalil Lab. Uh, so what did they do? The title is High Throughput Continuous Evolution of Compact Cas9 Variants Targeting Single Nucleotide Pyrimidine PAMs. Okay, so here's the abstract. Despite the availability of Cas9 variants with varied PAM compatibilities, some genomic loci, especially those rich with A's and T's, remain inaccessible by high activity Cas9 proteins. So it sounds like we have some Cas9 proteins that do work, but not high activity. Broadening the PAM sequence compatibility through engineering can increase off-target activity. That makes perfect sense. The Cas9 nuclease first checks for a matching PAM. So that's 3 bases. You can imagine after it checks for the PAM, it then checks if the short guide RNA matches up. So if you have a very broad PAM sequence, let's just say any 3 bases, it's just going to be checking all over the place, and it's going to have more off-target activity just because the sgRNA is going to have more chances to mismatch and allow the Cas9 to cleave. So you don't particularly want to just have a very broad PAM sequence. What they did was use directed evolution, which is an incredible tool in itself. Here's what comes to my mind when I hear directed evolution. I can't remember what the paper was that I saw, but you remove an essential gene for reproduction out of a bacteriophage. You put that gene as a plasmid inside of a bacteria, and you add an engineered gene for a specific molecular tool you're developing into the phage. So that would be the Cas9 nuclease that we're trying to develop pyrimidine-rich PAM sequence versions of. Somehow the phage more successfully accesses the reproduction gene that is inside of the bacteria as its molecular tool gets more effective. And then you run a small-scale survival of the fittest experiment. You basically do a brute force search of variants of the sequence of the gene or the amino acid sequence of the protein, and then you pick the best and you do variants of those, and then you pick the best and you do variants of those. That is the high throughput that they're referencing. Over time, random mutations make the tool better, and that version of the phage prevails. So you can basically use evolution in a directed manner. So using this strategy, they developed a Cas9 variant. They call it Nme2Cas9. Not sure what that stands for, but it can recognize a single nucleotide T or C. This right here does look similar to what I was imagining. So it looks like they did a couple different directed evolution circuits. They did one for just the new PAM sequence recognition, and then they tacked their adenine deaminase on the TATA and started optimizing for a base editor. I tried to go through like number or letter B here, and there are things that I don't know what they mean, like this cis-intine split G3. So yeah, I don't know what exactly. So like they're saying the accessory plasmid. So this is the AP, the blue ring. I don't know what that is. And I Googled that stuff and it doesn't tell me what it is. So I don't really know what exactly they did here, but it kind of looks like what I would think. But the bottom line is they're engineering Cas9 proteins to have more useful pan sequences, and yeah, that's good news. That's a bullish case. Also, I want to show you guys this thing real quick that David R. Liu tweeted a couple hours ago. Human TKTL1 implies greater neurogenesis in the frontal neurocortex of modern humans than Neanderthals. And that is a single A to G mutation in the gene TKTL1. So, yeah, that's pretty cool.